Sure.
So, hi everyone. Today we're welcoming Dr. Joan Bernhardt, senior scientist in the Geology and Geophysics Department at Woods Hole Oceanographic Institution. Um, so, I'm sitting in the Iowa booth in my exhibition, Object Permanence at Polka Gallery. The booth is plays on the design of an antique voting booth. And it's the Iowa booth. The Iowa booth is an educational space, a space to share ideas, a soapbox, a space to embrace our current time of Zoom life and bring that to the exhibition. Fun, it's currently functioning as a broadcast booth during the duration of the show. So for each event, it's going to look a little different. The canvas walls I'll be changing in and out. Um, so I just wanted to say that the voting booth has the connotation of being a political space. Tonight in Sharing Science, this booth is not political, it's a classroom. So it's a pleasure to welcome Joan tonight. Joan has many research interests, including benthic foraminiferal ecology and cell biology, which we're going to hear about tonight. I have a lot of gratitude towards Dr. Bernhard for being an inspiration and an ongoing collaborator. I've made tons of artwork um, learning about the uh, learning about the process of Joan's research. And special thanks to Laurel Von Rosk and Dr. Samuel Bowser for back in 2017 organizing an exhibition, Adaptation to Extreme. Several artists were included, and we were all responding to Dr. Bernhardt's research. I got to see Joan's passion and focus firsthand when I shadowed her and her team of researchers on a vessel, Gordon, the Robert Gordon Sproul, in the Santa Barbara Basin in 2018. It's interesting because I learned that Joan's focus is like artists I know, super focused, tireless, obsessive, and a dynamic thinker. That experience to learn about scientific inquiry and the tools involved has changed the course of my work. So, thank you, Joan. It's an honor to hear about Joan's research in forams today, foraminifera. I think you'll all be surprised to learn how important these tiny organisms are to understanding our world. Some of Joan's achievements include a fellowship at the Center for Electromicrobiology in Aarhus University, Denmark, a fellowship at Japan Society for the Promotion of Science. She was previously the Robert W. Morris Chair for Excellence in Oceanography at Woods Hole Oceanographic Institution, where she currently works and researches. The W. Storrs Cole Memorial Research Award through the Geological Society of America. And she was also a Fulbright Fellow at the University of Oslo in Norway. So please help me welcome a fellow Western New York native, Dr. Joan Bernhardt.
Thanks, Terry. Yeah, thanks for the introduction and thanks for the invitation. So I will just start sharing the screen here. So hopefully I can get this to work properly. Thank you for joining the Iowa booth. It's really fun to talk to Terry, and you'll hear about all our adventures over the next little while. So if any of you were on last night's lecture from Terry, he talked about this foram species that we named out of his artwork. And he says he doesn't know how to pronounce the name. Well, it's Samothonica anoxiki, and that means belonging to the sandy anoxic underworld. So you'll see pictures of this later, and it's the way I like to refer to our art science collaboration that we've had over the past few years. So I would like to, have any of you had, well, let's see, now I'm trying to go forward. Okay. Have any of you had a nightmare where you're suffocating? So I think a lot of people have, at least I have. So I'm not sure if everybody does or not. But it's important to know that all animals need oxygen to survive. And it's absolutely essential to human life. And um, oxygen is really necessary to all animals. Some people might argue that there are some that don't need oxygen, but in fact they do. And of course, this includes organisms living in the ocean. So at the beginning of this science project that we have, I wanted to do some art science work. And so we decided to go off on this adventure and think about from the perspective of suffocating, for example, and you'll understand the connection to oxygen shortly. Oops. So there are large areas of the ocean that have very little oxygen. In fact, some of the ocean, in fact, quite large areas of it lack oxygen altogether. So this map shows the areas of the ocean that have no oxygen at all, and that's shown in the white. Now, you might be a little alarmed because it looks like there's huge areas of the ocean that don't have any oxygen. But this projection or this map actually shows how much oxygen is dissolved in seawater at 300 meters water depth. And that's about 2/10 of a mile. So, so these are called oxygen minimum zones, or they have other phrases. And these areas are actually expanding. And this is alarming because there are organisms that live at 300 meters water depth. And some of them can live with a little bit of oxygen, but when the oxygen goes to totally zero, then those organisms have to migrate to an area where they can get oxygen. So those will go, most of them will go to shallower water depths, and then they're going to start crowding out the organisms that are living in the surface. And then you're going to have a lot more population of organisms, and they're going to use the remaining oxygen quicker. So it's like it magnifies a cycle where ultimately you end up with no oxygen. And so then, of course, organisms die. And these areas are expanding, as I said, and we call this oceanic deoxygenation. So it doesn't mean that the oceans are all going to go anoxic. The word anoxic means no oxygen. It just means that they're expanding. So an excellent example of deoxygenation you might have heard about in the news. This is an aerial photo of the region between the Mississippi Delta and Houston, Texas. And that darker area that you can see in the bay, more or less, that's, they call it the dead zone. And so what happens is you get a lot of nutrients coming out of the Mississippi River. And in fact, some of those are coming from Iowa, from all of the agricultural activities up there. And if you heard the previous dispatch from the Iowa booth, Sylvia she's a geographer. She was talking and showing a lot of old maps of the Mississippi River. In any case, we have lots of nutrients coming down the Mississippi, and then the ocean currents drag that water mass to the west, and it has no oxygen. And so you have huge die-offs of fish and other organisms that live on the seafloor. And so in the bottom, we have a graph over time that shows the extent of this dead zone. And you can see that it can be very, very large. And if you did statistics on this, you would see that it is expanding. There are some years where it's not as bad as others. And that's, yeah, we don't have time to get into all of that in today's talk, but it is a concern. So yeah, so there are some reasons for this deoxygenation, and the main thing is temperature. So we have global warming, and then there's some other things like the nutrient runoff. But today we're just going to be talking about the impact of low oxygen on organisms, and I'll talk about the microbes that we study. And these organisms are adapted to live in these conditions. So my collaborators are all shown here, most of them anyway. There's a graduate student and an undergrad from URI that I don't have on this slide. But so they've been on this adventure with us. And then our microbial collaborators are the foraminifera, or two particular species of foraminifera, actually. So forams, so you can call foraminifera forams, they're a kind of amoeba-like protist. So they have a single cell. Most of them are microscopic. And I'll talk more about them in a few minutes. But the picture that I show here, that's my all-time favorite species. It's called Nonionella stella. And you can see that it, well, there's probably about between 50 and 100 specimens here. And it has a little bit of a green color to it, blackish green, and you can see some white, that's its shell. So they make a calcium carbonate shell. So first I want to talk about where we sample. We sample off of Santa Barbara, California. And so Santa Barbara Basin, it's kind of a special place in the ocean, but there's other places that are very similar. I like to sample Santa Barbara because it's easy to get to compared to some of the other places that are off of Venezuela or the Black Sea, for example. So the basin lies between the mainland of California and the Northern Channel Islands. And it has a maximum depth of about 600 meters. So, you know, roughly a half a mile. And then it's basically a depression in the seafloor. So it's like a bowl in the seafloor. And so you have a combination of the local currents as well as chemical conditions in this area that cause the bottom of the bowl to basically be stagnant. And so it can go anoxic, or meaning have no oxygen at all in the deepest parts. And so that is, it's my favorite sample site of all times. It's really, really an interesting place. And this stagnation can last for well over a year. It can last for years, and sometimes many years. So this is a view of the seafloor in Santa Barbara Basin. The width of field of the large image is probably about 2 to 3 feet. And the white fluffy stuff on the surface is a particular kind of bacterial mat that it's a sulfur oxidizing bacteria. And so it's really quite developed in this particular image. And then the other image shows a core sample that we took from the seafloor at a similar time when the mat was similar. And so in the core, you can see those little whitish tufts. They look almost like little volcanoes. So that's what you are seeing on the seafloor. And you can see that there's a very thin rind of brown oxidized sediment but under that, the sediments are very, very black. So we have a very steep chemical gradient in these sediments. And it turns out there's a lot of very interesting biogeochemistry as well as microbiology going on in this area. So it's a natural lab that basically has been deoxygenated, if you will, for a very long time. And so we can study that to get an idea of what the ecosystem changes will be in the future as we have oceanic deoxygenation expanding over time due to climate change. So why would anybody want to study forams? Well, partly because they're beautiful. So this slide shows you 3 examples of 3 main types of foraminifera. The one that looks somewhat like a nautilus shell, that one is the shell is made out of calcium carbonate. So the same kind of mineral that is makes clam shells and snail shells. Then the one on the right is an agglutinated foraminifera. So that takes small sand grains or finer silt grains, for example, and it kind of glues them together with an organic bioadhesive. So it can literally glue the sand grains together to make its house, if you will. And then the color image is of a soft-shelled foraminifera called, well, Allegromids. It's a kind of a generic type or word for these ones that have an organic shell. So that would not fossilize, and it is just organics. And that picture also shows you some of the pseudopods on these, which are actually called reticulopods. They anastomose, so they can make like a net. And they're very good at using that for mobility, as well as some of them use it for food capture and for collecting sand grains if they're going to make their shell out of particles. So these forams are nearly ubiquitous in the ocean. They're both, they live in the water columns as planktonic organisms. They live on the seafloor as benthic organisms. They live all the way from the deepest parts of the deep sea in the Mariana Trench to mud flats. And also they live in high latitudes all the way down to tropical reefs. They're a major component in coral reefs as carbonate producers. And they can be very, very abundant on the seafloor or in waters. They have a very long fossil record. They evolved long before dinosaurs, before animals. And they have a very diverse fossil record, and there's also very diverse the living ones. So fossils have been used for hundreds of years, especially in oil exploration. And because they can be used to both date the rocks, so you know what age the rocks are, which is important when you're looking for oil and gas. And also you can use the species composition of all of the community and determine where you would want to go look for the oil or gas. They've also have been used more recently over maybe the last 100 years for geochemical proxies. So the ones that make calcium carbonate shells, we can analyze the chemistry of those shells, and we can determine what the oceanographic conditions were at the time that those shells were precipitated. So they're really, really important for understanding the past oceanic conditions and paleoclimate. And we need that information so we can better predict what's going to happen after we've had all this climate change that is ongoing. And finally, they are environmentally sensitive, so they can be used for monitoring remediation of pollution, for example. So here's an example of some of the diversity in these organisms. Everything on this slide, except for part of a staple in the lower right, is foraminifera. So you can see sometimes they're relatively large. The picture on the bottom right, that's from a reef flat in Japan. Those are called star sands. The original puka shell necklaces were actually foraminifera shells. The picture on the left is an assemblage from the deep sea. You can see a wide variety. A lot of them just look like spheres or long cylinders, but then you can also have some just beautiful ones. The one in the black and white image is another one of my favorite species because it has really unusual cell biology. And then there's one on the upper right that looks like a tree, and that literally is kind of structured like a tree, where it has a trunk and it has branches, and it puts these reticulopods out into the water column and grabs food. And it has roots also into the sediment. And that's about 3 centimeters, more than an inch tall. And that's from Antarctica. So everybody assumed when I was going to college that forams were aerobes, so that they needed oxygen. So while I was doing my PhD way back in the dark ages in the 80s, I went to Santa Barbara Basin and sampled there, and I realized that there were some forams living in really what I would call stinky mud. So it's not just anoxic, but it smells like rotten eggs. So then as part of my postdoc and early career, I went and did a systematic sampling along a gradient from where it's higher oxygen down to the lowest, stinkiest mud. And the species progressed over that distance. And so the ones at the top of the slide need more oxygen than the ones in the bottom. And the one in labeled J, that's my favorite species, the Nonionella stella, which I talked about briefly before. And it's really can do amazing things, as I'll tell you briefly about. So I started studying the cell biology of some of these, and I found some really interesting adaptations. Some of them are just unbelievable. And I'll highlight one. One is the Nonionella stella. You can see it here in this picture in the lower right. And it actually, what it does is it sequesters chloroplasts, which are the photosynthetic organelle that plants and algae have. And so photosynthesis, of course, needs sunlight, but Nonionella stella is hugely abundant on the seafloor in Santa Barbara Basin, where there is effectively no light, certainly not enough light for your typical photosynthesis. So when I realized this back many decades ago at this point, I always wondered how they could do it, and then what exactly were they doing metabolically. And so finally, the molecular methods got good enough where we didn't need huge amounts of material to be able to study these. And so I convinced some of my molecular biology colleagues to study these foraminifera. So this project that we are wrapping up now, we did some experiments. We went and got the species, brought them back, and then we manipulated the oxygen concentrations, as well as some other compounds like sulfide and hydrogen peroxide, which is a reactive oxygen species. And then we assessed the genetic response of these forams to be able to figure out what they were doing metabolically. And it turns out they are doing, we've already know that they can denitrify, and that's really unusual because most microbiologists think that only bacteria can denitrify, that they don't think that protists can do this, but we've shown that they can. And they have some, their denitrification genes, some of them are totally different than other genes. So they evolve these genes separately. So we have one manuscript submitted, hopefully to be accepted soon, and we have some more coming. So I'm not going to go into all of that kind of science. I don't even, well, I understand the molecular results, but I could never explain them really carefully to a molecular biologist. And so this is an NSF funded project. And so for NSF projects, they like us to try to convey our science to the public. And so when I was writing the proposal, I wanted to do, to use visual arts to be able to show how foraminifera are so unbelievably beautiful, and also to convey our science. So we were lucky enough to get funded. So at that point, then I needed to have, find some artists. So I reached out to my former postdoc advisor. I did a postdoc at the New York State Department of Health, the Wadsworth Center in Albany. So I have spent a number of years in the Capital District. I was there from 1990 to 1997. And so for those of you, you probably know what the Empire State Plaza looks like. And so we work there, or Sam used to work there, he's retired now. And so you might wonder where a research lab would be in the Empire State Plaza. Well, we effectively are underground. So we're kind of under the Corning Tower, which is the tallest tower. So if you look at the image on the left in the middle, we're basically, that's all of that block underneath the big tall tower. And those are not windows, actually, most of them. So it really feels like you're underground when you're working there. But there is one little atrium that you can see in the other view. And so we could see if it was snowing or raining outside. So anyway, I approached Sam about this, and he's the one that made that digital art in the second slide I showed. And it turns out he was married to Laura von Rosk, who is an artist as well as an art curator. And so Laura was recruited into this effort, and Laura and Sam recruited a number of artists, and these artists, I suppose I would describe them as science curious. And so over an entire year, Sam and Laura would have conversations with the artists. And so I provided a number of scientific images of foraminifera and their adaptations and the habitat and what have you. And then they would have conversations about the science and the relevance and importance. And then the artists went off and made some pieces of artwork. And I was working in Denmark at the time, so I was only part of, I think, one of these meetings. It was in the middle of the night for me, and they were sitting around up at Sam and Laura's house drinking cocktails while I was in the middle of the night wishing I was asleep. But anyway, it's really interesting. So that ended up being a multi-artist exhibition up in Lake George at the Lake George Arts Project. And this was a couple of years ago, actually. So there were, I think it was about 12 artists, and it was really interesting. I thought it was awesome. And the exhibition name was Adaptations to Extremes. So again, that's the picture of the Santa Barbara seafloor. And so that was the thing that all the artists considered was extremes, and how organisms, and in some cases people, can survive these exposures. So one special thing that we did with that was we had the artists write something about their artwork. And then we also wrote scientific perspectives on their interpretation and what they were had learned about the extremes that these organisms were facing. And so we have a compilation book that is online if you're interested to go look at it, and the link is there. But so here's an example without the prose, but the image that is called Science. So that's actually a relatively high magnification view of pseudopods of foraminifera. And then on the left was Eva Henderson's interpretation of that image. And so it almost looks like a roadmap, if you will. And then the other picture shows the actual part of the installation. And you can see there's a woman wearing a hat, which is made out of felt. And it's in the shape of an actual foraminifera. She made a number of foram hats. And in fact, she made my favorite foram into a hat. And this is my Nonionella stella felt hat. And these are the pseudopods coming out of it. So I'll spare you this for now, but I love it. It's absolutely beautiful. And the woman in the hat is looking up at a large piece, a large ceramic piece that is showing the view or perception of the view from inside a foram shell through the holes in its shell out to the environment that is extreme. So we had lots of different media types, and it was really interesting. Here's another example. This one was done by Charlene Leary. So if you think back to the picture on the seafloor and the core that I showed you, so she had seen these images along with this X-ray of the core to show you really the laminations or the layers. And so she made this sculpture which shows the laminations in copper tubing or copper circles. And then she put in microfossils, which were actually images from my scientific papers. I just thought that was unbelievably creative. So we also had a panel discussion. And so here's a picture of a number of the artists, not all of them, and then Laura and Sam as co-curators. And then of course there's Terry. So Terry was the only artist that actually came out on one of our collecting cruises. And he really, I have to say, it was transformative to Terry. He got in there slinging mud with us, and he got covered with seawater. He got sopping wet. And he also went around the ship and in the lab, and he visited us in Woods Hole. And he would find things. He'd go look in the garbage, basically, and he would collect things and old nets. And as he talked about last night, he would take our excess mud as well as some seawater, and then he would make ink for his prints. And so this is one of his printing presses that he made for this project. And if you haven't seen it, you can go and see some of these things in the exhibition. But this isn't, the actual press is actually making a print on the floor, you'll see. And he uses tin cans, aluminum cans that have been bent into shapes to make his prints. And then he delivers the ink through tubing to go into the different chambers. And so this is one of the pieces that Terry made. And he said, and it does actually look like foraminifera. And this is why we named it Samothonica anoxiki, belonging to the sandy anoxic underworld. And it actually does look like a foraminifera. This one is actually has a real scientific name, but you can see the grains of the materials are very similar in appearance as well as in shape. So this one can be range in shape from triangles all the way up to hexagons. So Terry had a one-person exhibition also in Woods Hole. And so he showed a lot of those prints that used the mud from Santa Barbara Basin, but he also did some additional printing methods there to show the forams. And this one, it's quite a large print, and it uses what I would call embossing. So it's there's relief to the print. And this is based on this scanning electron micrograph of a species from Santa Barbara Basin. And this one is really interesting scientifically because it has bacteria that live in the holes on the outside of the cell, but it's kind of in the little cubby hole of the pores. So they're symbionts basically. So that's really interesting scientifically. And then also, I think Terry has a few of his found aluminum cans that he has perforated. So basically, they're really very similar to what the forams can look like in the fossil record. And so, again, the perforations are effectively showing the holes in the foram. And these, of course, these pores are important because it allows exchange with the environment. So you can kind of think about them as window screens. So we're hoping to obtain some more funds for another collection cruise, hopefully, so we can go out and have some more fun with these fascinating forams. So with that, I'll turn it back over to Terry.
Great. Joan, thank you so much. That was really wonderful.
You're welcome.
So I would like to turn it over to the audience to see if anyone has any questions for Joan. And if you have a question, maybe type in the chat that you have a question and we'll call on you. Well, I'm going to start it off with a question.
Okay.
Well, let's see. So could you talk? I'm interested in a little bit more information about the fossil record, and basically you mentioned that the fossil record is useful for, it could even be useful for industries, right? And does that ever become a problem in the scientific community?
Oh, well, well, let's see, a lot of the people that learn about foraminifera, they end up going into the gas and oil industry. So they will become employees of Shell or Exxon or wherever. And then they will just, the oil companies, they drill all over the place and get samples, and then the data becomes it's proprietary data. So that will never get published. So that's kind of a decision of people that learn about these organisms, the fossil aspects. That's a decision. And of course, a lot of times they'll go and get a job and do that instead of do academia, where, we have other things that we do.
Yeah, that's interesting, because an industry that has a lot of funding figures something out, and then it's proprietary information.
Well, because the oil companies, they drill, they pay to get the samples. So they drill the holes to get the samples and all that kind of thing. Yeah. You know, when we do anything that is funded by federal sources, like the National Science Foundation, of course, we publish everything and put the data in archives, publicly available archives.
Thank you. So
Yep.
we have a question from Dawn DeShaw.
It's Michael again. Hi.
Mike-
Hi. I wanted to say I really appreciate the visuals that you were showing us of the small organisms. I was curious if you or a part of your team are responsible for those specific pictures, or are those more generic images that are available to scientists?
I would have to, you don't
It's
want me
okay,
to try to, I was going to say, you don't want me to try to share again, but anyway, I would say most of those images I took, I didn't take all of them, but
Can I
most of
follow
them.
that up with, like, what does it take to get those sorts of photographs?
Oh, okay. So the ones that are grayscale that showed the foraminifera shell, those are with a scanning electron microscope. So that's, yeah, I mean, it's kind of a common electron microscope in most universities would have one. And now for the past maybe 10 years or so, there's even quote tabletop versions, which are really literally the size of old desktop computers, a little bigger than that, but and they can take pretty good images. Yeah, so those where they look like they have relief, those are scanning electron micrographs. Then some of the pictures in there, the reticulopod picture that was taken by Sam Bowser, and that was with a light microscope. I believe that was, well, differential interference contrast, I think. I don't think I showed any transmission electron micrographs, but I certainly had showed them a lot, some of them to the artists, and some of them did artistic pieces based on those. And so transmission electron microscopes are also, most universities are going to have one. Yeah, some of those people that really pioneered that work, a lot of them have retired, and I have to say it's almost a bit of a dying art, because to prepare those samples, you have to prepare them in a particular way, and it's a science as well as an art to get good sections. You have to actually cut them with a diamond knife, and all of those people are retiring, like literally within the past few years. And I haven't done it for a while because it's I'm out of practice, so I don't do it anymore. So
Yeah, thank you. That's excellent. They were such beautiful pictures. They really helped bring to life what you were talking about.
thanks.
We have a question from Evelyn Pay.
Yes, hello.
Hello.
Hi.
I was wondering, Dr. Bernhardt, if you would talk a bit more about the forams within which within the pores of which the bacteria lived symbiotically. That I thought that was really interesting.
So just, sorry, you just want to know what the forams are doing with bacteria, like
Yes.
symbiosis?
Yes, exactly.
Yeah, okay. So that picture, that was also a scanning electron micrograph where they look like they were kind of on the top of a plateau, the bacteria. What I had done with that one was I decalcified the foram and then did a scanning electron micrograph with that. But that's the only species that we know of that does that consistently. But I have to say that not that many people have looked. So that one's really unusual. We don't know. So when I did a lot of this work, or when we did a lot of this work, it was before it was easy to sequence things with a very small amount of RNA or DNA. So we actually I have not sequenced that one, so I don't know what kind of bacterium it is. I would like to, and we might have a chance to do that. I'm starting a new project this summer, COVID permitting. It's funded by NASA, and we're going to be looking at some more species and looking at their genetic response. But there are a number of 4-am species that have symbionts, endosymbionts, so inside their cell. And one of the weird ones is where they sequester chloroplasts. So that's a little bit different kind of symbiosis. It's really rather unusual. There are sea slugs that do it, but they live where there's sunlight. So these 4-ams that do it in the deep sea are really, really unusual. And I call that the chloroplast conundrum. But there are other 4-ams that have more typical bacteria, rod-shaped bacteria, coccoid bacteria. And that's what we're studying now, is we're getting the genetic response from both symbionts as well as the 4-am. So you can consider the whole 4-am as like a community, but then we're also looking to see what the bacteria contributes and what the 4-am does for the bacteria, if that makes any sense. So it's really fascinating stuff. And there's lots of different examples. So there are many different types of endosymbionts, and we're just beginning to understand them. Like literally just the papers aren't even published.
Wow. Evelyn says, thank you very much. That's amazing. And we have a question from Sam. Sam, would you like to ask the question?
Well, I can just read it.
Okay, yeah.
So Sam says, can you
I
talk?
unmuted.
Oh. Hi, Sammy.
Joni, how are you? Hey,
All right.
can you talk a little bit more about denitrification? I know it's a huge discovery. Question,
Yeah.
exclamation mark.
Yeah. So for people that don't know, this is Sam Bowser, who was my postdoc advisor. And he also, of course, helped with this, the 8 expedition. And he and Laura are the reason that Terry and I met, actually. And so anyway, yeah, so denitrification is a way to break down some nutrients that come in, like river water from agricultural runoff and things like that. And so it was thought that only bacteria could break down the nitrate and return it to nitrogen 2, which is the main element of our atmosphere. And so a while ago, it was shown that some 4-ams, they showed that they could denitrify. There's many steps to the denitrification process. So it is still somewhat controversial. There are still some people that think, no, it's not the 4-am that's doing it, it's bacterial associates, whether they're symbionts or just bacteria maybe hanging on the outside of the 4-am. So we've done some experiments and some other things that it looks like the 4-am actually is doing the denitrification. So there just are the only other eukaryote or cells with a nucleus and organelles that do it would be fungi, and fungi are a little bit of an odd eukaryote, so they're not even really a protist. So anyway, yeah, it looks like these 4-ams actually are doing it, and some of these genes are very peculiar. And so that's what I've been working with the people at EORI. So Ying is a co-PI on the project, and she's a bioinformaticist who does modeling of metabolism. So, that is just stuff that I'm not equipped to do because my biochemistry is horrible. And so anyway, but we've found some really, really interesting results.
And then the other thing about it, Joni, is that the 4-ams are so darn abundant in the world's oceans that they are probably playing a major role in doing this.
Right.
Who would have thought? Who would have thunk?
Right. Well, so that's the thing, is that Yeah, in some cases, people have tried to do the, actually quantify their contribution. And so we kind of have some results where some people in certain places have found that the forams aren't really doing very much in terms of the percentage of the denitrification, and other people in other places have found a lot more. But I think as we study these more, we'll find out that a lot of them are actually doing a lot of it, a lot of the denitrification in places that we don't even think that they're denitrifying. Meaning they make like their own little habitat, and inside we have some evidence that suggests that at least Nonionella stella, my favorite foram that's green and sequesters chloroplasts, even if it's living where there is a little oxygen, so sometimes the basin will flush and you will have some oxygen come into the basin. But even if that's the case, that foram will stay doing anaerobic metabolism. So it won't switch to respiration. And so it basically just maintains its anaerobic activities, which is so this is something that hasn't been published yet. So it's pretty interesting. And that's also really, we're also getting some interesting insights perhaps into the early evolution or early diversification of protists. And so like I'm talking, this could have some impacts in the early Earth ecosystem back before you had animals. And that was some work that Sam and I did way back when. And, we think they were like the sharks of the Precambrian. Like they were the big bad boys that would go around eating everything. They were the apex predators. But that's another story for another day.
Joan, you make me want to come out of retirement.
Sammy, you should. Hey, you want to go to sea with me in July off LA? We just
Is Terry going?
No, not in this one. We don't have that much space. It's with an ROV, so we're going to get to use a remotely operated vehicle. And so, yeah, that's all, like I say, COVID willing anyway.
All right. Well, I'll try not to catch COVID. We'll talk later.
Yeah.
Thank you, Joni. That was great.
Thank you, Sammy. So it looks like Laura is writing something here. Laura, do you want to tell us what you wrote? So she's asking how much, so she says Terry has such crazy creative. Laura, hi.
There. Well, yeah, you could read my question. So I guess what I was getting at, I'm cheating here because I have 3 questions. So for Joan, did you ever worry about like having Terry interpreting your work? That did you just give him full like, okay, you run with it, Terry, you go? Or was it like, no, Terry, that's not it? Or was there anything like?
Well, and I think some of this also happened during conversations with the artists that you guys had over that whole year, where, I mean, sometimes Terry would ask a question and I'd say, well, that's not quite the way it works, or whenever I try to explain it. Sometimes it's difficult, and sometimes we just don't know the answer. That's the other thing, But I think Terry should answer that somewhat. But I would say I welcomed his totally different perspectives, and with all the other artists also. I mean, it's really interesting, and I would sit there and I'd say, oh my gosh, I never thought about it that way, or whatever. And it really is interesting to try to explain it in ways that different minds interpret things. Terry? You're muted.
Unmuted. Yeah, well, I would say that I would, I wanted to, I was given a certain amount of freedom, but I was also really excited about this opportunity to learn. So I had in my journal questions And I would often send off emails to Joan, sometimes when she was very busy. So it would be, she'd write, I got to answer this later or whatever, but like maybe a string of questions. Um, so I think it was, um, I, I was maybe sort of careful um, and to misinterpret things, but I also, in some cases, like the idea of the metaphor of what some of the organisms might be doing. Last night, I described them as maybe an attribute. So inventing or making a version of a foraminifera, but like highlighting one of their ideas, or like the idea of agglutination, say, just like running with that idea. Because I think that these printing presses are agglutination, and collections of stuff. Um, but I can remember one, the being out on the ship at sea. It was really highly scheduled. There was a lot to do, but I remember one really nice moment that we got to sit and talk, and I just got to ask questions and interview Joan. And I think I feel really fortunate to just get to absorb all that, because I think those are the moments that kind of sparked the ideas for making the work. So yeah, so thank you, Laura, for your question.
Thank you for that. That's great. I love that. I love hearing about how you guys work together. Okay, I'm gonna mute myself.
Right. Well, it's a little bit after 8, so I kind of just want to finish with a little question that is related, unrelated, Joan.
Mhm.
And I'm curious about when you knew that you wanted to be a scientist.
Oh, yeah. Um, well, I remember that there was a spelling quiz in 3rd grade, and I did not spell the word science properly. And I remember that I broke down into tears. I went to the teacher in tears that I couldn't spell the word science right. And she said, well, you've told me that you want to be a scientist, so you better learn how to spell it. So apparently in 3rd grade, I already knew I wanted to be a scientist. And then in terms of when I wanted to become, I've always been fascinated with water. So well, Terry alluded to the fact that we both grew up in Western New York. So, but I would spend summers on one of the Finger Lakes, and I learned to swim when I was something like 3. And but I also remember one time when I was learning how to water ski, and I had fallen, and a big fish came up, and he was checking me out while I was waiting for the boat to come back, and I just got really, really scared. I think it was probably a little perch, but at the time, I thought it was this enormous thing. And so then I kind of got very curious about what lives down there in the murky deep. That was fresh water though, so, and then I guess when I got older, I realized the ocean was a lot more extensive, and there was probably an awful lot more unknown things there that would be interesting to study.
That's really exciting and inspiring. So Joan, thank you so much.
Thank you.
Thank you everyone for attending tonight's talk. So next week there'll be a sort of a concert performed by Tommy Santa Claus at 7 o'clock from the Iowa booth, and he'll be playing some of the instruments that I made. The following week, which is March 4th, Dante Hayes will be giving a lecture. He's a ceramic artist. And then March 11th, Brian Dewan will be performing some music. And so all this is on the Apulco website. So the link was just dropped in the chat. So I hope you'll come back. And once again, thank you, Joan.
Thank you. It was great.
All right. Have a good one.
Thank you, everyone.